So just a little introduction. Bacterial DNA analysis involves examining the genetic material of bacteria. This can either be genomic or plasmid DNA. So we do this in order to understand the structure, function, see if we can find any new genes or try to understand the functions of those genes. So that's the importance. We can also use it to understand genetic makeup and understand the role of these genes in the environment. So this has many applications in research. For example, we can try to understand virulence, which is how a bacteria is able to make people sick. Antibiotic resistance, which is an important thing. We need to understand if these bacteria, for example, are resistant against certain antibiotics. So just an overview of the whole process from extraction to comparative genomics. So first of all, you need to extract the DNA, of course, so that you have material to analyze. And then you're going to sequence that DNA, and then you will do some quality control and read trimming, and then sequence assembly, and then you can do gene prediction and annotation, and finally comparative genomics. I will be going through each one of these. So the first step, which is usually done in the lab, is extraction of the DNA. First of all, you need to prepare your sample. So you collect your bacterial cells from a culture, for example, where you had grown them. And then you will collect the cells from this culture by centrifuging. So the cells are going to settle at the bottom of, let's say, your Eppendorf tube, and they will look like a small pellet. And then after that, you can break apart the cells, which is cell lysis. You can do these chemically, mechanically. So there are different methods. So for example, chemically, you can use SDS, which is a common chemical used in the lab for cell lysis. Mechanically, you can do these by sonication, which involves using sound to break apart the cells. And then after this, you can purify your DNA because it will have some impurities once you have extracted it. And then the sequencing part involves, first of all, library preparation. So before you sequence, you need to have a library. So this is made up of your fragmented DNA, which you obtained. You can also do amplification, which involves increasing the amount of DNA because bacteria have very small DNA, so you might want to do that. And then after that, you can sequence. So this means you read the DNA fragment so that you know the bases that make up the DNA. And then afterwards, you will obtain your raw sequence reads in formats like FASTQ, which is the most common format. And then now the analysis part, which is the bioinformatics part that I am more aware of. The lab part, I'm a little rusty on that. Okay, so the first analysis you want to do is to do quality control and read trimming. So quality control uses tools like FastQC, and the purpose of this is to assess the quality of your raw sequence reads. You want to check if they came out nicely or not. So this takes in FastQ files as input, and this is the command that you will run. I'm just gonna see if we can try to use okay, yeah. So this FastQC command is what you will run on terminal, and then this will output a file in form of HTML, whereby you can go and open it and check the quality. So that usually shows things like the Fred score, how good your reads are, and all that. So if you find that you have some low quality reads, which happens usually at the beginning of the DNA sequence, you can do trimming. So this involves removing low quality bases and adapter sequences. So adapter sequences are sequences that are used in library preparation. It's usually the opposite of the template strand. So it's like replication, but it's synthetic. And then, so the tool used here is Dreamomatic. You can set parameters for quality threshold, for example, Q30, which means how much you want to trim. So the higher you set the threshold means it will be very stringent. If you set it a bit low, it will be less stringent, so it will trim less of your DNA, your sequence reads. And then you can set the minimum read length to trim. So if you, depending on the sequencing method you used, which are usually long read and short read sequencing, you can decide the minimum read length. So if you used long read sequencing, then you will definitely have to have a longer read length than if you used short read sequencing. And then this tool or program takes in paired and FASTQ files, and then it will output for you trimmed FASTQ files. And then after that, if you're feeling confident with your reads, with your sequences, you can progress to your next step. If you're not, you can do quality control again, using FastQC to check the quality and repeat the whole process again. So this is the command you run on Dreamomatic. So you add the input file and the output, the input, reverse read and forward. So reverse means it's the opposite strand, and then forward means it's the actual strand. Sorry, my biology is again a bit rusty, but yeah. And then you will run your command and it will output for you the trimmed reads. And then this is where you add the parameters that we discussed earlier, such as the stringency that you want the program to use while it's trimming your reads. And then sequence assembly and assessment is the next step. So the whole purpose of these is to have a fully assembled genome, because at this point you only have fragments. So we want to rebuild the whole genome as it was initially. So this uses tools like Spades or Velvet, and then it will take in the trimmed FASTQ files as input, and then it will output for you the now fully assembled contigs, and it will give you a file that is in FASTA format. So the command that you run here would be this. So you have to put your forward reads and then your reverse as well, just like in the Dreamomatic command. And then you have to specify where it should output the output files for you. And then after doing the assembly, you can evaluate the quality of the assembly using Quest. So this checks metrics like N50, total length, and the number of contigs. So N50 is the quality of your assembled bases, if I am not mistaken. And then it takes in assembled contigs in FASTA format, so this program, and then as input, and it will output for you a report, just like in FastQC, and then you can review that report to see how well Spades or Velvet or any other program assembled your genome. And then after that, now you can do gene prediction and annotation. So gene prediction, we want to find out what genes are there in my genome. So this uses tools like Procar, and then you will input the FASTA file that has your assembled genome, and then it will output for you, sorry, just a second. It will output for you an annotated genome file. This is usually in the format like GFF or GBK. So again, that is the command that you will run, specifying your output directory and your input file. So this enables us to get information like gene names, the position of that gene on gene on the genome, and the coding sequence. So that is the components of the GFF file. And then next you will do annotation. So this means you want, now that you know what genes you have, you want to find out their functions. So you can use a database like KEGG or Gene Ontology. And then the tools that are used include BLAST2Go, which usually uses the Gene Ontology database, and it has an interactive UI. So here you don't need coding knowledge. So BLAST2Go will first read your sequences, which you have to import. So as FASTA file, and then you will display, it will display for you a table of all your sequences in that, in your sequence file, sorry. And then after it has displayed for you this sequence table, you can perform a BLAST search. You can either select all sequences on the table or certain sequences that you want. And then, so this BLAST search is usually dependent on like the type of sequence. You can blast a protein sequence or nucleotide sequence, but for this purpose, we're blasting nucleotide sequence. So the type of blast search that is performed is a BLAST X. And then you will choose the database that you want to blast against. So this means it has to like run a search. It's like a search for your genes against the database. So you can, at this point, choose NCBI or another database that you desire, like UniProt, if you're doing if you have protein sequences. And then you have to set an e-value. So this e-value shows you the significance of the match, the likelihood that your sequence matched another sequence in the database, and not by chance. So if it's lower, it means it's better. If it's higher, it means that has a lot to do with chance. That might not be the right match. And then after you have performed your BLAST, you can do mappings to assign gene ontology. So gene ontology terms are terms such as the biological process the gene is involved in. So let's say like DNA replication, and then the location of the biological process, so like the cytoplasm or the mitochondria, something like that. So this is assigned based on the BLAST hits. And then after that, you can configure some annotation parameters. So annotation parameters, there are several of them. So there's annotation cutoff. So this is the score that is used for cutting off the GO terms, well, assigning the GO terms, sorry, to a sequence. So a high cutoff means only high significant hits will be used for annotation. So what this means is that only those sequences that are close, that have a closer match will be used for annotation. So if you have a lower cutoff score, it means you will have more false positives. And then a GO weight. So this is weighting of a GO term. So before the assigning of a term, let's say before we conclude that this gene is involved in a certain biological process like metabolism, we need to have a weight. So that is what determines, so that we cannot have a gene that is involved in something like replication being assigned a biological process in metabolism. And then the HSP hit coverage cutoff. This is the minimum coverage for your high scoring segment pair. So a segment pair is the match between your query sequence and the sequence in the database. So minimum coverage means how much you want the match to stretch out for. So like we can decide we want the match to stretch out for like, just an example, 5 base pairs, 10 base pairs. So that's the coverage. And then setting these to 0 means that all your pairs will be considered. So that is what you want. You don't want some of your base pairs to be left out. And then after that, you can start your annotation process, and the output of these will be displayed to you on the Blast2Go UI. So here we have to visualize this in order to understand what annotations have been made or what conclusions have been made about our sequence. So for these, Blast2Go will provide you with a graphical, it will provide you with graphs or graphical tools that will help you visualize the distribution and relationships that it's obtained for you. So some common graphs that Blast2Go will display for you is geo-term distribution graph, and another one will be an annotation pie chart. So the first one, it will show you the distribution of geo-terms within your database. So this is usually categorized by the process the gene is involved in, the molecular function within that process, and then the cellular component. So this will just help you understand where most of your genes lie and their locations. And then the annotation pie chart will be a summary of the geo-term distribution graph. So it's basically just almost something similar, but more condensed. So here is an example. So you can see there is a more graphs here. I just discussed in the previous page. So we have number of geo-terms identified by length. So this will just show you how many of these terms were identified for which lengths of your sequences. And then the geo level distribution graph, this shows you the number of annotations against the geo level. So the geo level can be 3. The first level is usually the general biological process. And then the second level is the more specific subcategories. So first, we just start by generalizing. For example, this is a metabolic process. We don't say which metabolic process. And then second, we go to the now a bit more detail. We specify, for example, this is a carbohydrate metabolic process. And then the third level is usually now more specific. So it will tell you something like, this is glycolysis, which is a type of carbohydrate metabolic process. So this is just to help you understand how your genes are distributed in different levels. And then for the, sorry, I don't think I've left anything. Yeah, for the geo terms by length graph, which I discussed first. It's just the genes of different length bins and their count. So you can find multiple genes being assigned one or more geo term. So the shorter the gene, the lesser the terms it's assigned, because we are limited in finding out how much it can do. And then the longer the gene, the more variety it will have. And then after that, you can export your data in a format like CSV. And then you can go to the next step, which is comparative genomics. Sorry again for the repetition. I am not sure why my PowerPoint does that. So comparative genomics, as the name says, is just comparing one genome to another. So the reason for these is to help us understand evolutionary relationships and find out if we have any unique genes. So to do this, you first do sequence alignment, which is aligning your sequences, as it is in the name. And these will use tools like Clustol Omega to align your sequence. And then this is an example of a command that you can run on your terminal. So you specify the input file, which is in FASTA sequence, and then you specify the type of output file or the name of the output file that you want this program to use. And then it will output for you the aligned sequences. After you get these aligned sequences, you can feed them to the phylogenetic analysis tool. An example is Mega, and it will use that sequence alignment to construct for you an evolutionary tree. So an evolutionary tree just shows, it's like, it just shows how far or how closely related your bacteria are. So if they're closely related, then the branches will be a bit closer. And if they're not, then it should be farther. It's like a family tree, but for evolution. And that's the end. You have analyzed your first bacterial genome. I apologize for the little hiccups. If anyone has questions, I will be taking them now.
Okay, thanks so